So I want to thank Joan and AZBio for the opportunity to speak to you today and introduce you to cancer prevention pharmaceuticals and talk to you briefly about some of the clinical trials that we are currently running. Let me see which one. So the mission of cancer prevention pharmaceuticals is to really prevent cancer or the recurrence of cancer by identifying specific risk factors in different patient populations. And we're really modeling this on what has been done very successfully in cardiovascular disease. In cardiovascular disease, they were able to identify and develop diagnostics to really target those patients that were at high risk for developing cardiovascular disease. Following that, they then implemented a variety of preventative therapeutics. That was combined with various lifestyle modifications such as diet and exercise, and really resulted in a significant decrease in deaths that are associated with cardiovascular disease over the last 30, 35 years or so. And what we would like to do is use this same paradigm in cancer and preventing recurrence of cancers or preventing cancer in those patients that are at high risk. As you can see, there's only been a very minimal decline in the cancer-related deaths in the last few years, and we'd really like to see a bigger impact over time. So CPP, or Cancer Prevention Pharmaceuticals, we're a small, late-stage clinical drug development company based in Tucson, Arizona. We were founded based on some technology from Dr. Gene Gerner at the University of Arizona. We also have received funding from a variety of sources, several of which are a number of Arizona venture groups, such as TRAC and Desert Angels. We also have received funding from other biotech angel groups. We have a partnership with Xeria and Tillot's Pharmaceutical Company, where we have licensed the technology for development in GI indications in both Japan and in Europe. And then we have other funding opportunities. We have a partnership and a collaboration with the National Cancer Institute, which is supporting one of our 2 phase 3 trials, which I'll talk about in greater detail in a minute. So what are the goals of CPP? We would really like to target inflammation and genetic changes that are really the drivers for the development of precancerous lesions and cancer. And we'd like to do this through a variety of ways. We'd like to do it through some pharmaceutical interventions, as well as through developing diagnostics and companion diagnostics, which was just touched on in the last session. We're really targeting a few key drivers. In this case, we're going to focus predominantly for my talk on polyposis and GI cancers, in particular colorectal cancer. And we're targeting 2 key factors, ornithine decarboxylase, or ODC, and the resulting polyamines that are generated from the ODC gene. And these are driven by a number of key genetic factors and mutations that occur predominantly in colorectal cancer, such as the APC mutation, KRAS, as well as others. And what normally occurs, and you have to show some really nice colon pictures, especially after lunch. So you start with a very nice, healthy, normal colon mucosa. What will happen through these genetic changes or inflammation, sometimes diet will also play a role, is that you get the development of these adenomas. And adenomas can be precursor lesions for cancer. These will eventually progress where you get full-blown cancer of the colon. As mentioned this morning, designing these prevention studies, if you're looking at a normal, healthy population, can take tens of thousands of patients and decades to perform. So what we focus on at CPP are patients that are at high risk for developing cancer. In the context of colorectal cancer, we look at those that are colorectal cancer survivors. Those upon screening are shown to be polyp formers, since those are the precursors for colorectal cancer. Those with GI disorders where they might have gastritis or robust inflammation and chronic insults to the colon that can result in further genetic changes, as well as patients where there is a genetic susceptibility to colorectal cancer, such as patients that have familial adenomatous polyposis or HMPCC. We then take that population and we are trying to put them through a funnel where we can increase screening or implement new companion diagnostic and diagnostic tools, identify therapies that can really target the specific risk factors that these patients may have, and implement that with lifestyle changes so that we can really reduce the number of deaths from colorectal cancer. And this is really key. As you all know, colorectal cancer is very prevalent in the US. There's about 150,000 patients that are diagnosed with colorectal cancer every year, and 50,000 patients that will ultimately die from colorectal cancer. We would like to implement some effective prevention therapies to decrease these numbers significantly. We are being very ambitious, but we like to have good goals. Decrease the number of deaths and implement early screening to really make a difference in this area. So our lead compound we call CPP1X. It is an irreversible inhibitor of the ornithine decarboxylase gene. And as you may recall from a few slides ago, this is one of the key drivers of growth, tissue repair, as well as carcinogenesis in the colon as well as in other indications. We have a number of different programs that we at CPP are either running or we are doing in collaboration with groups such as the NCI or other investigator-initiated trials. We have our pivotal phase 3 trial is being run by CPP in familial adenomatous polyposis, or FAP. I will talk about that in greater detail in the next few slides, but this is an orphan indication where they are genetically predisposed to get colorectal cancer. This trial is being run in the US as well as Europe and Canada. CPP1X in that study, as well as in the second large phase 3, is being evaluated in combination with the NSAID Solendec. The second phase 3 is the collaborative study with the NCI and the Southwest Oncology Group looking at colon adenoma therapy. This is the PACES trial, or phase 3 trial. This too is in combination with Solendec, or CPP1X with Solendec, and I've got a few slides to touch on that as well. Then we have a number of investigator-initiated trials in a pediatric indication called neuroblastoma. There's about 600 to 700 kids every year that get neuroblastoma. This is a disease of the nerve cells in kids, and there's a really nice genetic mechanism as to why CPP1X may have an effect in this population. In these investigator-initiated trials, the compound is being evaluated both as a single agent in patients that are at high risk, as well in combination with standard of care therapies. And I would like to note that one of the clinical trial sites for the NMTRC consortium is actually Phoenix Children's Hospital. We also have a number of other programs and typically orphan indications where these patients are at high risk for developing colorectal or other diseases. We're looking at Lynch syndrome, which is also known as HMPCC, with some key investigators in Europe that really focus on this disease. And then secondly, we also are looking at gastric cancer, and this is a collaboration with investigators at Vanderbilt University. And they recently received NCI funding for a phase 2 trial evaluating CPP1X versus placebo in patients in Honduras as well as in Colombia. So I'd like to spend the next couple of slides telling you a little bit about FAP and our FAP 310 clinical trial. FAP is an orphan disease that affects around 30,000 folks in the US and 40,000 in Europe. It is an orphan indication as a result. Most of the patients, around 80%, there is an inherited risk and inherited mutation in the APC gene that occurs. There's another 20% that have spontaneous mutations that are also drivers of the disease. Given the genetic mutations, these patients will develop at very young ages hundreds, if not thousands, of polyps within their colons. As a result, they have a 100% chance of risk of getting colorectal cancer if they do not have any interventions implemented. Typically what happens with these folks is that they are families of FAP patients. They will get a number of symptoms. They will then undergo annual, if not more frequent, endoscopies to evaluate polyps and so forth. Typically by late teens, early 20s, these patients will have their colon and or their rectum actually removed. And that's not the end of the intervention. This is a lifelong disease that results in frequent endoscopies, the whole GI tract. They have multiple surgeries, they get desmoids. It's really a horrible disease that has significant impact on quality of life, as well as impacts on morbidity and mortality. This is, aside from surgery, there are not a lot of pharmaceutical interventions that are available for this patient population. And as you know, as I noted, these patients are at very high risk of still getting colorectal cancer. So we really wanted to make an impact within this orphan indication. We have a pivotal phase 3 trial, which we call FAP 310. This is a 3-arm randomized blinded trial where we are enrolling 150 patients. They will receive either CPP1X plus the NSAID Solendec, CPP1X plus placebo, or Solendec plus placebo. They will undergo treatment for 2 years, and we are looking at a time to an FAP-related event as an endpoint. Currently, this trial has enrolled 81 patients across 14 active sites. We have another 6 sites that are pending enrollment. These sites are within Europe as well as US and Canada. We anticipate the trial to complete enrollment by the end of this year, such that we can complete the study by the end of 2017, file with the FDA with the market approval targeted in late 2018, 2019. So really want to make an impact on this orphan disease. We're making great progress, and we're excited about what we're doing in this indication. The next trial is the phase 3 colon adenoma therapy, where we're looking at prevention in patients that are at high risk for developing adenomas. This is a collaboration with the NCI and the Southwest Oncology Group, and it's called the PACES trial. There are over 500 IRBs that are currently approved. We have 45 subjects that are currently randomized. This is a very large trial where we're looking at around 1,400 patients. The trial design is actually on the next slide, where we're taking patients that are stage 1 through stage 3, a year after post-resection. Then they'll be randomized to one of 4 arms. The design is very similar to the FAP trial, but there's one additional arm in that there's a placebo arm. But you have the combination of CPP-1X plus Solandac, CPP-1X alone, and Solandac alone. Patients will be treated for 3 years, and then the primary endpoint is a colonoscopy at the 3-year time point looking at high-risk adenomas or secondary colorectal cancers. And then there will be a 5-year follow-up. This is a very large trial, around 1,400 patients. But we do have data from a previous phase 2B, phase 3 that was done by Dr. Gerner and Dr. Meskins out of the University of Arizona and University of California, Irvine, that showed very promising results. So overall, Cancer Prevention Pharmaceuticals is focused on developing prevention therapies and diagnostics for high-risk patient populations. We're utilizing some very strong mechanism of action and well-tested scientific basis from previous trials in the development of CPP-1X to develop this compound. While we are a small company based out of Tucson, we do have a large worldwide team that are helping us develop these compounds through a number of different trials, including the NCI, key academic centers, Sanofi, as well as others. And our goal is to really take these late-stage clinical trials, which are initially in colorectal cancer and colorectal cancer-related diseases, but expand to other orphan diseases and unmet needs, such as neuroblastoma. And with that, I think I will actually finish on time. So thank you.
I think the discussions, there's so many